paper I'm gonna present is titled "Strengthening the Kimball Indirect Pathway Promotes Resilience to Compulsive Cocaine Use." So, cocaine addiction is now a very severe social problem. The cocaine is widely used as a kind of party drug. They use a lot in clubs or street gangs. But this cocaine has problems when chronically used. It can cause increased risk of strokes, or since it increases the blood pressure, it can cause harm to your heart. And well, just like what kind of problems can occur. So people are studying how can we make people less addicted to cocaine. And so people get addicted to cocaine because it works as dopamine, serotonin, and noradrenaline reuptake inhibitor. So it keeps these, especially the dopamine, keeps in the synaptic cleft for a very long time, and it used cocaine, so you feel very good at that moment. But when you do cocaine for a very long time, then you get problem with this receptor balance, or so your dopamine receptor gets internalized, so you don't have enough dopamine receptors, or sometimes they get just not sensitive enough for as before. So most of the studies are done at the ventral tegmental area, or nucleus accumbens and prefrontal cortex in the mouse study. So when you see some news, there are some celebrities using cocaine, and some just can't stop doing cocaine, but some people can stop doing it and get back to the normal life. So this study, this paper is about what makes the difference between the resilient individuals and non-resilient individuals. So the study focused on the role of D1 as medium spiny neurons and D2 medium spiny neurons. D1 medium spiny neurons express dopamine D1 receptors, and D2 expresses dopamine D2 receptors. D1 medium spiny neurons forms direct projections to midbrain, so it's called direct pathway. And D2 medium spiny neurons form indirect projections to midbrain via pallidum and subthalamus nuclei, so it's called the indirect pathway. The activation of the direct pathway increases the locomotion and serves as a reinforcer for some like food or drugs. And activation of indirect pathway decreases the locomotion of the animal. So in this study, they used the behavioral study in the first. They made this chamber, and whenever there's a light signal coming from this correct signal, the light makes a nose poke into this hole, and then they get the intravenous cocaine infusion. So they did this training for a very long time. They did it over 6 to 7 weeks.
How much cocaine?
It depends on the individuals. So they checked the perseverance and motivation score. The perseverance is checked by the mean number of nose poke during drop-off period. So it's like when you're doing computer and the computer just freezes, you continue to click on the mouse until it gets fine. So it's like that. So when the cocaine just stops, when you just stop giving cocaine to the mouse, some individuals just try to get cocaine, so they keep doing the nose poke during the drop-off session. So they check the mean number of nose poke, and motivation is they did the test where you have to make more nose poke to get the cocaine in the next session. So in the first, you just poke one time and they give you cocaine. But when you keep doing it, like you have to do like 50 times or something. So they check the motivation by counting the number of nose poke they done when they get the last cocaine. So the higher the nose poke made, the higher the motivation. So they check whether the amount of cocaine they finally got had a correlation with this perseverance score or motivation score. In perseverance score, they didn't see any correlation between the amount of cocaine individual got and the perseverance score, but in the motivation, they found the correlation between the total cocaine the individual got and the number of nose poke they made until the final cocaine. So it just makes sense because the higher motivation score means that you get more times the cocaine. So, and they made the combined behavior score using these two types of behavioral score, and it still had a correlation between the total amount of cocaine they got. So they divided these individuals into two groups, and it was divided by the 10 milligram per kilogram of cocaine the mouse got, because in this parameter they showed the difference in locomotion. So the individuals which got higher than 10 milligram per kilogram had higher motivational score, and the other didn't. So with this, after this, they checked if they could see the difference in alpha and MD ratio and the D1 MSN neurons, because in the previous studies, it was already known that individuals with cocaine had higher alpha and MD ratio than D1 MSNs. So they saw that in D1 MSNs, they had higher alpha ratio, but it didn't significantly change in D2 MSNs. And they wanted to see if this change was about some specific spines or it was just a general change. So they did the two-photon uncaging study, and they saw that in some set of D1 and MSN spines, it had a higher alpha NMDA ratio, but some others remained same, so it showed a shift to the right. So they concluded that the synapse that had input from the cocaine use showed the higher alpha NMDA ratio. But as you can see, there are some individuals showing higher alpha and MJ ratio in the D2 MSNs. So they divided this group with so they saw that if they could get the difference between animals which have alpha and MJ ratio difference in D2 medium spiny neurons by dividing them with behavioral scores. So the individuals with so they saw that the behavior score had the negative correlation with the D2 MSN alpha and MD ratio, but D1 MSN didn't show that. So they wondered if they could increase the D2 MSN glutamatergic signaling, then they might be able to make the individual resilient to cocaine. So they used the GRED technique to modulate the D2 MSN activity. So this is the scheme of GRED technique. So in this study, they used the HM4-DI receptor, and if you give the mouse this cyano compound, this receptor activates the GI pathway, and it activates the BERT channel, so it just shut down the neuron activity. So they use the D2 specific pre-line mouse and this H4-MI receptor, and they also use the channel rhodopsin to. So they infused the mice with these two kinds of virus and the NEC with 1-to-1 ratio. So they gave the laser in this nucleus and cubus to activate the D2 MSNs, and they measured the iPSC in this VV region to see if there were any change in the signal coming from the nucleus and cubus region. So they could see the iPSC decrease when they treat CNO in this mice. And in the behavioral study, when they give the CNO this mice, those mice show higher motivational score to cocaine than the selling group. So this might implicate that decreased D2 MSN activity causes higher motivation to cocaine. And they also checked the motivation for food rewards with the same scheme. And they said they couldn't see any difference in this food reward.
Which food is used for that? Pellet? Just pellet?
They said just pellet. But the N number was only 3, and these graphs are not totally same. So
The reason I ask is in the general science paper, some authors shows that D2 is also important for binge of the binge means eat a lot, 혹시 in Korean. So D2 is also involved in binge, so I think I asked that.
yeah, but I also thought this food reward behavior study was not very reliable because it didn't use much animal enough. So they moved on to the optogenetic stimulation study. So they infected the D2 MSNs with channel rhodopsin, and they gave the blue light during the cocaine self-administration behavioral test. So in this graph, you can see very clear that when you give the blue light, the mouse shows very decreased attempt to get cocaine. So these black bars mean the attempt to get cocaine, and the red bars means that the cocaine was actually given. So they show about 70% decreased total rewards were given to this optogenetically activated mouse. So they said that this D2-MSN activation decreased this mouse motivation for cocaine. So in the study, they saw the individual mice showed variability toward cocaine consumption, and the alpha ratio was increased in D1-MSN after cocaine consumption. And in some resilient individuals, the D2-MSN alpha ratio after cocaine consumption had increased, and GRID-mediated D2-MSN inactivation caused higher motivation toward cocaine, and optogenetic activation of D2-MSN decreased cocaine-seeking behavior. But I think some more studies should have been done to say that D2-MSN surely have the effect on the cocaine consumption. So my first question was, why do resilient individuals have higher alpha-MD ratio in D2-MSN after cocaine consumption? Because it wasn't just the innate difference from the beginning, because they didn't see this difference in the sham group. So it occurred after cocaine consumption. So the cocaine consumption for some individuals made this difference. And is there, in the study, they saw the postsynaptic mechanism of higher synaptic transmission in D2-MSN. So we can study if there's any presynaptic mechanism of potentiated glutamatic input into D2-MSN. And there can also be some downstream target study of D2-MSN and cocaine resiliency. And I didn't put this on this slide, but in this study, they didn't mention about any locomotive activity of mouse, but as this result might be the result of decreased locomotion of the mouse, so they need to check that kind of problem. This is the end of my presentation, so if you have any questions.
Is there any discussions in the science field that this kind of resilience in cocaine is somehow similar in kind of act like a aversive? This direct and indirect pathway, there are many studies that are saying it's acting as a reward and aversive signal. Does that explanation and this drug resilience or motivation kind of explanation have some kind of
Yeah, there was mention about that point in this paper also. So, it said that so that's why they did the study of food reward. So they wanted to show that this wasn't just only about the reward and the reward for some getting the reward stronger, but because they didn't show any difference in food reward, so they wanted to say that it is just a cocaine specific effect. But I don't know, as Taehyun said, that there might mentioned about this because it's that the reward for food is like acting so strong, so they might so the activity of D2MSM might not have that big, might not cause the big difference in food reward studies. If there are no